Stanford University.
Today I'm going to talk a little bit about myself and how I ended up doing work at Jasper, and a little bit about the final outcomes of that research, but most importantly about undergraduate involvement and how integral of a role that was in the quality and the quantity of data that we ended up collecting, and that's now multiple peer-reviewed publications. So this is a photo of me as an undergraduate back in August 2007. And the summer before, so the summer of 2006, I had done a summer research program down at Pepperdine University with Professor Stephen Davis. And he really instilled in me a healthy appreciation for chaparral vegetation. So he would take us out, we had to learn all the names of the plants, and did a pretty extensive field study. I found out later that he had been a postdoc for Professor Harold Mooney. And when we came up to Stanford 1 year later, we're here for the Ecological Society of America conference. And at the end of the conference, there was a one-day field trip to Jasper Ridge, I'm sure part of one of their many outreach programs. My professor was completely jazzed to take us to Jasper Ridge and show us, where he had done some of his work, as well as we got the chance to learn about the global climate change experiment. And I'm not sure exactly what Nona is telling us here, but I look extremely interested in whatever she's saying. But talking about, finding the wonder and enjoyment in just being out in nature, after I left Jasper Ridge that day, I was hooked. I was like, I'm going there, I'm doing work at that field station, I'm going to Stanford. It was the only place I applied for graduate school. I definitely had my heart set on doing work there. So fast forward a couple of years later, when I met with Tad Fukami and we talked about potential project ideas, and one of these projects was a new project that he was starting up at Jasper Ridge, I, of course, jumped on that opportunity. And not only was it really exciting to be able to do work there, and Jasper Ridge is such a valuable resource because, like you've heard many times today already, it's so close to campus, and science is the kind of thing where you will make a mistake, you will fail. You do need to redo those experiments, and if your field site is halfway around the world, that's a little bit difficult. So having such a world-class field station so close is really helpful in going back and repeating things and trying new things when you learn new information, but also in doing continuous research over a long period of time that not a lot of people have the luxury of doing. So all of that was definitely a plus. And then I had a very strong background in molecular biology, and this project involved looking at microbial communities. And being able to marry field work with molecular lab work is not something that a lot of science can do. So being able to do both of those things at the same time, I was completely excited and hooked, and I love taking risks, as Nona said, so I was all about it. So now I'm just going to talk a little bit about the study system. Many of you have been up at Jasper Ridge, are docents, and you know a lot about it already. But I'm also going to talk about what we found. So I think now that I can look back on my whole thesis, which I just submitted 2 days ago, I would say that the main core of it is looking at the biogeography of microbes. So as we've heard from multiple researchers today, for naturalists since forever, since Darwin, since before Darwin, have been thinking about biogeography, which is where do we find organisms in ecosystems, and how does that change through space and time? And that has been well cataloged for macroorganisms, for plants and animals. But as far as microorganisms go, well, we can't really see them, first of all. And then when we can see them, when they're in certain formations, how are we able to tell different species apart to know what they're doing? And then taking that one step further, as we humans change the environment, how are we affecting these communities? And how are we affecting the ecosystem services that they provide to us? So for instance, I think I heard somewhere that humans have like 2 pounds of gut microbes that we need to digest our food. And plants form very tight associations with microbes in their roots, and they need these microbes to access nutrients in the soil. So they're very important to the functioning of ecosystems, but we know very little about exactly what they're doing and where they're located and how that is changing. So this system provides us with a very neat way to look at this. One of the limitations of microbial communities is that they are hyper-diverse. So if you drill like 1 centimeter into the soil and you try to look at what is there, you're going to have decomposing microbes. You're going to have microbes in association with plants. You're going to have free-living ones, pathogenic ones. So the question is, what are they doing, and how do you sort of parse all these many different species apart? So this system is interesting because we're looking at microbes that live in the nectar of flowers. So flowers are initially sterile, and the sugar content is so high that not everything can live in there. So it really filters out down to only a handful of species. And then these microbes are being vectored from flower to flower by different pollinators, so on the birds of bees and on the bodies of birds and bees. So another question is, what effect are these microbes having on these pollinators? The plant-to-pollinator relationship is well established, but what role is this third party playing in this? And for instance, we found that bacteria that live in nectar, some of them, reduce the pH so much that they make the nectar acidic and it's not as palatable to pollinators. So in that case, that flower might be less likely to be pollinated, having an effect on the plant and how much fruit it produces. So as you can imagine, looking at thousands of microbial communities and looking at surveying hundreds of flowers takes a lot of time and man-hours. And of course, if you need man-hours, you go to the undergrads. And so initially, it started out with just a few volunteers in our lab. So this photo is of Grace and Sophia, the 2 undergrads, and Diamantis in the middle next to Bill is another graduate student that was helping out at the beginning. And of course, the wonderful docents, Bill in particular, I see he's holding one of the old eye button holders that I heard are now upgraded. And so just having undergrads there to help collect a lot of the samples and do really intensive data collection, so counting many different flowers, loading temperature data, was extremely integral to the story that we were able to tell. And then we also had the Bio 44Y class, and Pat Seawall really helped coordinate that as well. That basically mobilized 100 undergrads into the field, marking and collecting data. And something that was really fun for me was, one, being able to work closely with my advisor in the field. I don't think a lot of graduate students necessarily get to work that hands-on with their advisor, so that was wonderful for me. But also being able to work closely with the undergraduates and seeing them get very excited. And they figured out pretty quickly that we had no clue what was going on and that we were learning as they were learning. And so when they brought their data to us and they're like, oh, look what we found, we were like, oh, yay, that's like super exciting. Do you want to continue going down that road? And so when I told you about nectar pH a little bit earlier, that idea actually started from one of the undergraduates in the class. So we were measuring things like water availability or temperature. And one undergrad was like, oh, could we measure a little bit of the nectar chemistry and see if maybe that's changing? And we're like, okay, pH strips are pretty cheap, why not? And that goes from that idea all the way into a full-scale thing that multiple people in our lab are currently working on and that has been published. So I think that's really wonderful. So I'm just going to show a little bit of what we found. And in my attempts to make my science translatable, I have learned to use Photoshop this week, so you guys get to be subject to my Photoshopping skills. So what we found, so the students did all the flower counts. And what we found is that as flower density in a particular location increases, you find more microorganisms in those flowers. So if you have more flowers in one giant bunch, then the percentage of those flowers that have been colonized by these microorganisms is higher. And then if you look at separate patches, so even if the patches had the same amount of flowers, if you were in a patch that was next to a patch that was already colonized, then you'd be more likely to be colonized. And if you were in a patch that was further away, and closer to another patch that was not colonized with these microorganisms, then you were less likely to be colonized. And so this makes me think of a few things. One is sort of an infection, like disease model. So if you live in a big city crowded next to a lot of people and a virus outbreaks, then you're probably more likely to get that virus. Or even if you live in a small city but everybody around you has the virus, then you're more likely to get it. And we're seeing something very similar here. And so that means that this is really dispersal limited. And when you think of why organisms are found in particular places, the first thought you have is their environment. So if they're well adapted to a specific environment, then that's probably where you're going to find them. And then the second thought you have is maybe competition. So if something else is better adapted than they are, it will beat them out and they won't be found there. But in our case, maybe those things are playing a role, but the real driver is, are they able to get to that place in the first place? So dispersal is the #1 thing driving this. And we wanted to look at the pollinators themselves, but we were having a hard time catching a lot of birds up at Jasper Ridge. And so my collaborator, Chase Mendenhall, who's one of Gretchen Daly's graduate students, was like, I catch thousands of birds in Costa Rica every year. Let's take this study down to Costa Rica. And so that's another theme that I've sort of heard today, is not only doing great research at Jasper Ridge, but also taking that research out into the world and translating it into sort of wider themes. And so we repeated this study in Cotobruz, Costa Rica, but we also looked along a gradient of human land use. So we looked at some areas that were located in coffee fields, and then we looked at some intermediate forest fragments, and then they have a smaller reserve there. So we also put some plants there. And so we're able to see not only are humans changing, since humans have transformed this environment, how is that changing what we see in these microbial communities? And we're also able to take a closer look at the pollinators themselves. And so because there is no one flower that sort of grows along all these gradients, we used artificial flowers, which was basically sort of Eppendorf tubes popped into felt caps, filled with filtered banana nectar. Exciting. And what we found is a very similar trend to what we found up at Jasper Ridge. So those fake plants that were in areas that were forested, and so that had a lot of other flowers and a lot of other plants around them, were more likely to have microbes in them. And then the areas that were less forested, had less forest cover, were less likely to have microbes in them. So the coffee fields were less likely to have microbes than the forest reserves. And then no birds at all visited our plants, which is very sad, but nocturnal moths did visit. And those plants that were visited by these nocturnal moths were also more likely to have microbes in them than the plants that were not visited. And so this is sort of telling us very clearly that this is mediated by the disperser, and that the dispersers, so the pollinators, are attracted to forested areas. And so this has some implications for agriculture. So we know that microbes are either pollinators are either more or less attracted to flowers with specific microbes in them. We also found distinct species of microbes. So the forested areas had a very different signature of these microbial species than the non-forested areas. So who knows, maybe in these agricultural fields, there are the species that are less palatable, so that could mean that there are less pollination services being provided to those flowers that are in coffee fields versus those flowers that are in forested areas, or vice versa. But I think the overreaching theme here is sort of that dispersal is driving this, and we were really able to start this project and get a real sense of how to do this work at Jasper Ridge and sort of take some risks and then transfer it out onto a much wider scale along this human land use gradient and get a better sense of what was happening out in the real world. And so with that, I just want to acknowledge some people, especially all the undergrads and the Bio 44Y students. We even had some high school students working with us. And Nona, thank you so much, and Bill Gomez as well. Thank you.
That was really interesting, and I like this project. So I just have a question that goes to what would be the information that you're gathering, what would be the possible applications of this, of what you're finding out and understanding here in any broader way, shape, or form?
So the wonderful thing about basic research is there has to be no applications, or there doesn't have to be. But I would say that as we look at this further, we know that these microbial communities are depleting some of the resources in the nectar. So they're changing the amino acids, they're drawing down the sugars. So one possible thing is they're either more or less attractive to the pollinators because of that. We don't know exactly, well, I shouldn't say that. I know Kabir has done some work on this, and so has Rachel, a current postdoc in my lab. So we have some sense of which species are doing what, but then another step is sort of figuring out how are the pollinators responding to that change in nectar composition, and I think that's ongoing research that's currently happening. So all of this is still being pursued and developed. Yeah.
What are you going to do next?
It's like the most stress-inducing question, so I'm going to ignore it. Anybody else? I don't know. That's the short answer.
I have a question, actually. I'd be really interested in hearing what you think your research tells us about how we think about ecosystems and natural systems in general. In other words, as you look at these, because as Nona said at the beginning, these have become sort of like model systems.
Yeah.
What are they model systems of?
Oh my. So as far as plants and animals, there are, unifying theories of biogeography. And so I think right now people are sort of trying to develop the same thing for microbes. And there's a very healthy debate happening. One theory is sort of, okay, these microbes are pretty much everywhere. And it's more or less how stringent the environment is can depend on whether or not they can live there. And we're seeing that that's not necessarily the case. So I think going a little bit further, I think it's really trying to answer the questions of what are the driving factors. So is it like the environmental conditions? Is it dispersal? Because they can disperse much further than plants and animals can, for instance. So I think sort of going in that direction, it's really trying to understand why we find them in certain places. And my work has shown that it is a dispersal limitation thing, but we're finding different patterns depending on what organisms these microbes are associating with. So I think it's just a really interesting sort of burgeoning field at the moment, and I'm excited to see where it goes next.
We started out this morning talking about Jasper as the birthplace of the theory of coevolution, and one of the possibilities that your work raises is that it's a whole different kind of coevolution where the microbe is actually influencing the pollinator success or the pollinator choice or that kind of thing. And do you see this as having the potential to kind of represent coevolution involving more than just two parties?
Oh, definitely. And I think, so with plants and animals, you can obviously see what's happening. You can make those observations. But I think now that we are able to identify microbial communities, I think we're going to be seeing a lot more of them in established mutualisms or established co-evolutionary relationships. So I think, yes, I think it's definitely, you know, sliding in there as we get to understand what's happening a little bit more. I hope my committee does not ask me questions this broad. Just saying.
Just remember, you've been videotaped.
Oh my.
One last question.
We will ask you.
So you were working with undergraduates and had a lot of other people helping you in this. How do you think this experience will influence your research in the future?
Okay, so the first question is whether or not I'm doing research in the future. That's number one. So I'm trying to understand, your question is how has my interactions with the undergrads influenced me? I think more so than thinking about the future, it has really made me nostalgic for my past. So I've had wonderful mentors along the way that have really put me on the path to where I am now. And so in interacting with them and in seeing them getting really excited and sometimes, you know, cutting that string and being like, no, you're going to have to figure it out for yourself. All of those things remind me of the wonderful experiences that I've had with my mentors, and I hope to be that for them. So that's more of the way my brain works when I interact with them. Thank you.
For more, please visit us at stanford.edu.